Alright, welcome back to the next video in our sequence on population genetics and microevolution. So, in the previous video, we talked about Hardy-Weinberg equilibrium and how it can be very useful to estimate the allele frequencies and genotype frequencies within a single population. And that's important because we said that that's the basis of determining whether microevolutionary change occurs within a population over time. But I kept hinting at the fact that Hardy-Weinberg has limitations because it makes a number of assumptions that are not always going to be true. So that's going to be the first thing we talk about in this video. What are those assumptions, and for what reason are they not usually in line with reality? So the idea of Hardy-Weinberg is that for a population, you can assume that that population is stably in equilibrium if certain assumptions are true. Now, what are those assumptions? Let's go through them. Number one, you assume that there are no new mutations over time, and in other words, this means that there will be no new alleles introduced to the gene pool appreciably over any period of time. So if the population 100 years ago only has 2 alleles for a particular gene, then 100 years from then, or 200 years from then, or 1,000 years from then, you should still just have those 2 alleles. There should be no mutations that cause a 3rd or 4th or 5th allele to pop up. So we are assuming that no new mutations will occur over time. And of course, that is not really going to be a good assumption, especially if you're tracking populations over a very long period of time. So the next assumption is that there will be no gene flow, meaning that there is no migration of individuals into or out of the population. The only new members of the population should be those that are produced via sexual reproduction among members of the already established population. So you shouldn't have any newcomers coming into the population, possibly bringing in new alleles or a different frequency of alleles, and you can't have any members of your population get up and leave. That would cause the frequency to change as well. This is possibly the most erroneous assumption, and we will talk about why later on, but Hardy-Weinberg assumes random mating, meaning that there is no sexual selection, there's no selection of partners or mates based on any sort of criteria on fitness or physical appearance. Basically, this assumes that if a population has 50 members in it, and if one of them is male, it is going to choose its female mate, or vice versa. The female might choose the male. The female would choose its mate totally randomly. So it would be a totally random proposition. The female would not be choosing its male mate based on any sort of criteria other than eeny, meeny, miny, mo, essentially. So this one is absolutely hardly ever going to be true, and we will talk more about why later on. We are also going to assume no genetic drift, so there's going to be no change in allele frequencies or genotypes due to chance events that really have nothing to do with natural selection. So no genetic drift. And we will more closely and carefully define genetic drift and provide examples of that later on as well. And then finally, no selection. So no evolutionary pressure being placed on the population as a whole to adapt to anything. No major changes in the environment. No introduction of new predators. Nothing that places pressure on the population to change their allele frequencies and come up with new possibly beneficial adaptations and physical characteristics. So obviously natural selection is going to play a big role in microevolution occurring. So if there is no pressure, if there is no selection occurring, then allele frequencies absent genetic drift should not be changing over time. So these are the 5 assumptions of Hardy-Weinberg, and as we are going to very carefully discuss later on, none of these assumptions are particularly good. So you are hardly ever going to find a population that in reality is truly in Hardy-Weinberg equilibrium. But Hardy-Weinberg is still a very useful tool to use, as we will see. So Hardy-Weinberg can still be useful in determining whether microevolution has occurred by treating a fictional population that is in Hardy-Weinberg as being your benchmark for, okay, this is what would happen if no microevolution has occurred, and we do have a stable population. And then if you collect your empirical data, your own allele frequency and genotype frequency data, And if they don't match the Hardy-Weinberg theoretical data, then you can usually use that as a pretty good indicator that some sort of microevolutionary change has occurred. And basically, the magnitude of how far off your numbers are from the predicted theoretical Hardy-Weinberg numbers, that should be pretty reflective of the degree to which microevolution has occurred within that population. Okay, so we said that these conditions are hardly ever met, so if we look at several examples of these conditions and we provide some specific real-world examples of, well, why would it not be true? So for the random mating condition, we said that, well, that's usually not going to be true, because usually what you're going to see is what's called non-random mating. So in non-random mating, your individuals in the population are probably going to pick their sexual partners based on some sort of criteria, whether it's the black moths only mating with other black moths, or some sort of sexual dimorphism, which we'll talk about in the third video of this chapter. There's usually some basis, whether it's physical or more abstract, on how we pick our partners. We don't usually just do it randomly. So because of non-random mating, you are not going to see random assortment of alleles as a result of sexual reproduction. And what that's going to result in is a change in genotype frequencies over time. So if two black moths only ever mate together, then you are going to increase the frequency of homozygous dominant individuals within the population, and you're also going to decrease the frequency of heterozygotes within the population. And since that leaves the homozygous recessive individuals to mate together, since the black moths won't mate with them, then you are going to see an increase in the frequency of homozygous recessive individuals. So the heterozygotes become less frequent and the two homozygous become more frequent. But this is not enough to say that microevolution has occurred. In order for microevolution to occur, you have to see a change in allele frequencies, not just genotype frequencies. Okay, so then let's look at the case of no selection. So we're assuming there's not ever going to be a selection pressure placed on this population. Well, that's probably not going to be true, because the longer the population exists, the more likely that something about their environment is going to change for the worse for them. So in this particular situation, you have a predatory bird show up, and let's say, for the sake of argument, the bird has kind of a hard time seeing the black moths contrasting against the dark background of the bark of the tree, but maybe the bird has a really easy time spotting those white-peppered moths. So the selection pressure in this regard would be the presence of the predatory bird, and the adaptation is having that black body and wing color that makes you harder to spot. So over time, the presence of this selection pressure would change the allele frequencies, make the dominant alleles more frequent because that's the adaptation necessary, and make the recessive alleles less frequent. So yes, microevolution would occur in that case. So the condition of having no mutations, if you do have mutations, which, again, the longer the population exists, the more likely that becomes, that is going to add new alleles into the gene pool, and just on the basis of how the math works out there, as we saw in the previous video, that is going to change allele frequencies too. So if you have a new allele pop up in the gene pool, you are going to definitely have some members of the population that take on some new, unique physical characteristics. So yes, microevolution does occur, but not necessarily because of natural selection. We can't say it's natural selection unless we prove that that evolutionary change is adaptive in nature to some change in the environment, some sort of selection pressure. So we talked about the condition of no gene flow, so no migration in or out of the population, but of course that is going to occur every now and then, and when it does occur, you're going to have individuals carrying alleles into or out of the population, and more than likely that is going to change allele frequencies, but again, that is microevolution, but it's not necessarily because of natural selection. And then finally, you're assuming no genetic drift, which usually makes the assumption that you have got a very, very, very large population, and we will learn when we talk about genetic drift here in just a minute. Very large populations are a lot less susceptible to the effects of genetic drift. So in reality, a lot of populations are actually very small, and they actually are a lot more susceptible to genetic drift. So whether it's the founder effect or the bottleneck effect, we will more carefully define those here in just a little bit. But this will cause some changes in allele frequency just because you may lose members of a population just because of dumb luck or some random chance event. Okay, so let's look at 3 different examples of what's happening to this population. So there's 3 different examples. You have the top one, the middle one, and the bottom one. So the top example is what would occur if this population is in stable Hardy-Weinberg equilibrium. So in one generation, we'll call this the F1 generation, you have allele frequencies that we calculated before, 0.2 for the dominant allele, 0.8 for the recessive allele, and then you have the clearly defined genotype frequencies that we also calculated before. So the idea here is that if all those conditions are true, that mating is random, there's no selection, there's no gene flow, there's no mutation, there's no genetic drift, the population is very large, then after a generation of breeding and producing offspring, even with those offspring being new members of the population, everything should still be exactly the same. The allele frequencies should be the same, the genotype frequencies should be the same. That should be true as long as all of those assumptions are still correct. And as we are going to see, they are not always correct. So let's look at this middle example here. This middle example considers a situation in which sexual selection occurs, in which, say, the female decides she's only going to mate with a male that has exactly the same phenotype as her. So this would be a case of black moths only mating with other black moths. So as we saw before, if this is the only assumption that is not true, then that really should not change the allele frequencies over time. But what it is going to do is it's going to change the genotype frequencies over time. So you can see both of the homozygous genotypes become more prevalent, so they've both increased by 6%, but the heterozygote frequency has gone down by 12 percentage points. So that is the natural consequence of choosing your partner based on phenotype. If you're dealing with a gene, a trait that only has 2 alleles, then the heterozygote becomes less and less frequent, but this still does not indicate that microevolution has occurred. Like we said, in order to make that declaration, we have to see a change in allele frequencies, and here we only see a change in genotype frequencies. And then in the bottom example, if we actually see a case of selection pressure being placed on the population like we see with that predatory bird, then we should see a change in both allele frequencies and genotype frequencies. So since the bird has a lot more success finding and eating the white-peppered moths, we would expect those recessive alleles to start to disappear from the gene pool because those white-peppered moths are not surviving long enough to reproduce and pass their genes on to their offspring. So here you actually see the allele frequencies totally flip. Now it's 80% dominant alleles and 20% recessive alleles. And we see that impacted in the genotype frequencies too. Now 64% of the population after the introduction of this predator are homozygous dominant, and only 4% are homozygous recessive. You can see the heterozygotes are unaffected. So in this particular case, we would say that microevolution has occurred. This is an allele frequency change that is due to adaptation due to natural selection because of the presence of a selection pressure in the form of a predatory bird. Now, of the ones we've talked about, this is probably just about the only one for which we would say that natural selection is the cause of microevolution. But in other cases, such as with non-random, or such as with gene flow or genetic drift, those are changes in allele frequencies that are not necessarily due to natural selection because there was really no selection pressure being placed on the population. Okay, so let's talk a little bit more about these assumptions. We've kind of convinced ourselves that most of these assumptions are probably not going to be true for any given population, but let's delve into this a little bit deeper. So genetic mutations, so we'll first consider that assumption that there's never going to be any new mutations within a population, so no new alleles being made. This is important to consider because genetic mutations are the raw material for evolutionary change because they introduce new alleles into a population. And if you have a new allele, you have a new possible genotype that an individual can have, and that can possibly lead to new physical characteristics that may end up being adaptive later on when some selection pressure makes itself known. Now, what we're going to see here, and something that I think we've talked about before, is that not every organism, not every species of organism accumulates mutations at the same rate. So some are faster at it than others. So mammals like us, we tend to be very slow at accumulating mutations. We are not rapid mutators. So we actually get most of our genetic diversity because of meiosis, through independent assortment and crossing over. But as we've clarified before, meiosis only shuffles the distribution of already existing alleles. It doesn't do anything to make new alleles. So in order for humans like us to make new alleles, we have to do it through genetic mutation, but that process is rather slow. Now, in contrast, if you consider prokaryotes like bacteria, like that video you watched at the beginning of chapter 1, or at the beginning of this first video of this chapter, bacteria tend to accumulate mutations very, 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 very fast. But they have to do it that way because that is their only method of introducing genetic diversity, because they do not reproduce sexually with meiosis. They reproduce asexually with binary fission, and that, just like mitosis, produces genetically identical offspring. So the only way bacteria and prokaryotes have to introduce genetic diversity into a population is with randomly accumulating mutations very quickly over time. So when we think about adaptation and mutation, mutations can really fall into one of three categories. And the reason I want to bring this up is because mutation and the word mutant are words that are kind of ingrained in our vernacular, and I think there's kind of a common misconception about exactly what mutation means. So if you'll remember, a mutation is just a change in the genetic code. A mutation doesn't necessarily mean that you're going to sprout a second head or grow a third eye or sprout extra fingers or anything like that. A mutation is just a change in the genetic code. So based on that knowledge, mutations can fall into one of three categories. You can have a good or adaptive mutation, which creates a new allele that imparts some physical characteristic that is actually good and necessary for surviving some sort of selection pressure or some current environmental challenge. You can have a mutation that is detrimental or bad, in which the new allele imparts some kind of physical characteristic that makes survival surviving to reproductive age more difficult. So a good example of this would be the kinds of mutations that cause us to get genetic diseases like Alzheimer's or Parkinson's or things like that. And then finally, probably the most common kind of mutation is one that is neither good nor bad. So one that you are probably not ever going to notice. So in this particular case, the new allele neither improves nor impedes survival in the current environment, but there's still the possibility that there's something about it that, if you continue to propagate it within the gene pool, maybe some new environmental challenge or some new selection pressure comes along, and now suddenly that neutral mutation is actually adaptive and good, or possibly detrimental and bad. It's just kind of hard to say. Okay, so let's talk a little bit about gene flow. So gene flow, of course, is the movement of alleles into and out of a population. And usually gene flow will occur between populations that are geographically isolated, but some aspect of the environment still makes migration of genes possible. So if you look at this example right here, you've got two different species of pea plants, so very similar to the type that Gregor Mendel worked with. So you've got two different types of pea plants that are growing in fields that are separated by a big river. Now, what that means is that since these plants are nowhere near each other, they can't actually reproduce with each other because they're separated by a geographical boundary. But if spores or seeds or pollen is picked up by, say, a bird or a bee, or if the wind picks something up, and that gets carried over the river to this other population over here, then that is going to allow for the sharing and intermixing of alleles and the gene pools of these two populations. And these two populations will grow closely together, and it makes it unlikely that they will ever experience a speciation event. Or in a second example, if you want to talk about something that's not plants, breeding individuals may be able to migrate into or out of a population based on their own choice, and there may be something about them that allows them to overcome some sort of geographical boundary, whether it's a bird being able to fly, a fish being able to swim, people being able to walk and climb and run and things like that. So those are good examples of gene flow not only being possible, but being a lot more likely than not. So as I kind of mentioned before, continuous gene flow between two closely related but separated populations slows down the progress of divergent evolution and speciation. So as long as two populations still are experiencing gene flow, it makes it very difficult for them to ever get to the point where they become so different where they no longer interbreed. But if you do get to the point where gene flow is no longer possible because of some irreconcilable geographical boundary or something like that, then speciation rates really will pick up quite a lot because the two populations are totally separated and they no longer interbreed. So it becomes a lot more likely that if you did get them back together after a period of time, they probably wouldn't be able to breed anymore. Okay, so now let's talk a little bit about genetic drift, something we have not really provided any examples of yet. So we saw before that genetic drift is going to be an event that causes allele frequency changes that are really not because of natural selection, but rather because of probability and basically dumb luck. So there are several different types of this, so we'll talk about the bottleneck effect here on the next slide. So what you're looking at here is another bottleneck effect, but I have a kind of a better example on the next slide. So a really good example of a genetic drift event is something like a natural disaster. So if you're looking at this particular situation here, our initial population has 10 frogs in it. So you can count them up and count 10 frogs. The trait that we are considering for this population is the skin color of the frog. The frogs can either have green skin or they can have brown skin. So you can kind of look here and kind of consider the phenotype frequencies. 10% of the population is brown and 90% is green. So what you'll kind of notice here is that this population is rather small. There's only 10 members of the population. And as we said before, having a small population makes that population very, very, very susceptible to genetic drift effects. Okay, so let's say that we have some kind of natural disaster, like we have a major flood, and the river waters rise, and that washes away 5 of the green frogs. So now if you look at your population after the natural disaster, there's only 5 frogs left, and one of the frogs still happens to be brown in skin. Well, because of how the numbers work out now, 20% of the population now has brown skin, and 80% has green skin. So if all you were doing was considering the allele and genotype frequencies here, you would see that those things changed over time. And if you didn't know any better, you would want to make the assumption that there was some sort of adaptive phenomenon going on here where having the brown skin was good and adaptive and allowed that frog to survive over time, and there was something bad about having the green skin. When in reality, that had nothing to do with it. It was just dumb luck that it was 5 of the green frogs that got washed away. The 5 frogs that got washed away, it could have just as easily contained that brown frog. And if that had occurred, we would be coming up with a very different conclusion. We'd be saying the brown frog, there was something about having the brown skin that was bad and unadaptive. So the whole idea here is that genetic drift effects produce changes in allele frequencies that really have nothing to do with anything other than just dumb luck. It just happened to be that way because of a chance encounter. As we said, genetic drift effects will be especially strong in small population, and a big assumption that Hardy-Weinberg always makes is that you have such a large population that even a genetic drift effect is not going to change your allele or genotype frequencies over time. So one of the most common occurrences of genetic drift is what we call the bottleneck effect. This is a case of genetic drift that involves a random event preventing the majority of individuals in the population from entering the next generation, like what you see with a natural disaster. A good way to illustrate this effect is literally with a bottle that has a neck. Who would have thought it, right? So look at this situation here. We have a glass bottle that contains a number of marbles. These marbles come in 4 different colors: blue, green, red, and yellow. If we count up all the marbles that are in the bottle to start with, we can see that 53% of the marbles are blue, 26% of the marbles are green, 13% of the marbles are red, and 8% of the marbles are yellow. Now what we're going to do is we're going to pick up this bottle and we are going to pour a small sample of those marbles into a glass beaker here. We're not going to pour all of the marbles, just some of them, just a small fraction of them. So we can assume that each marble represents 10 alleles in a population, so that's going to be 5 individuals within the population. And we're going to assume that pouring out alleles into this glass beaker, that's going to represent your population that survived some natural disaster or some natural phenomenon. And then the marbles that are left in the bottle afterwards are going to be those that did not survive, and they will, of course, disappear from the gene pool. So if we take a peek at what is left over, what got poured into the bottle, we can see that the allele frequencies have changed extremely drastically. So the green allele has increased in frequency by 18%. The blue allele has decreased in frequency by, it's increased in frequency by 8%. The red allele has decreased in frequency by 2%. And the yellow allele has totally disappeared from the gene pool. So again, if you didn't know any better and you were just looking at the beginning and the end, you might come to the conclusion that, well, there must be something about the color of the marbles that affects their likelihood of surviving some sort of natural disaster. There must be something about these alleles that affects how likely you are to survive, when in reality that is not the case. So the alleles, the marbles in this case, that were most likely to get poured into the beaker were those that just so happened to be on the top of the pile. So you can see that it's mostly blue, red, and green marbles that are at the top. The yellow marbles just happen to be towards the bottom. So the bottleneck effect here is that there is some aspect of the design of the bottle that makes it less likely for the bottom marbles to get through and more likely for the top marbles to get through. It's not because of color, it's just the random chance of how these marbles were distributed in the bottle to start with. So another way of thinking about the bottleneck effect is that after a bottleneck effect genetic drift event occurs, your next generation is going to comprise of individuals and their alleles that really just so happen to make it just because of dumb luck and random chance. So what you see here is that disproportionate random sampling produces essentially a founder effect, which we talked about before, and this drastically changes allele frequencies even though no selection has occurred. And that is the most important thing about genetic drift. You see allele frequency changes, but it is not because of natural selection. It's because of dumb luck and probability. Okay, so if we want to talk one more time about the founder effect, this is a case of genetic drift in which a new population is started from just a few individuals from the previous population. So the alleles that get carried over, again, are really just determined by random chance. It doesn't necessarily have to do with any sort of natural selection or selection pressure. The new population that gets founded from members of the original population, it's just dumb luck what frequencies of alleles they happen to carry with them. So the new population is almost certainly going to have different allele frequencies than the original population. So this is especially going to be more pronounced in new populations that tend to be very, very, very small. Just like we saw with the assumptions of Hardy-Weinberg, that we always assume that a Hardy-Weinberg population is going to be very large so that it becomes immune to genetic drift effects. So consider this example here. Let's say that we have a population of 10 individuals, 5 double A, 3 heterozygotes, and then 2 little a. So 5 homozygous dominant, 3 heterozygous, and 2 homozygous recessive. So the allele frequencies for this population would be capital A is 0.65 and little a is 0.35. So let's say that 2 of the heterozygotes and 2 of the homozygous recessive individuals get up and leave and they found a new population. So if they do that, then the allele frequency of their new population would be very different. It would be capital A is 0.25 and little a is 0.75. So what you see here in this new population is you see that the allele frequencies have changed very drastically. And because these individuals got up and left, the allele frequencies of the original population are now very different too. It's not because of natural selection, it's just another case of genetic drift. It just so happened to be these members of the population that got up and walked away. Okay, so now let's talk about the idea of non-random mating, which, as I said, is probably the most ridiculous and non-realistic of the assumptions of Hardy-Weinberg. So, as we said before, most organisms do not choose their mates randomly. Most individuals choose their mates assortively, meaning that they choose their mates that have largely the same phenotype for some given characteristic or maybe characteristics. So we saw that before in the case of, well, maybe the black moths only want to mate with other black moths, and that would leave the white-peppered moths to only mate with other white-peppered moths. So what this implies is that individuals will reject mates that have differing phenotypes compared to their own. So if you're dealing with a trait that is governed by Mendelian dominance, this is going to do exactly what we said in the previous video. This will increase the frequency of homozygotes, and it will decrease the frequency of heterozygous individuals within the population. You can go back to that specific example that we looked at in the first video and see that that absolutely is true in the case, in that middle case where we do see sexual selection occurring. So different things can kind of influence mating behaviors other than assortative mating and sexual selection. Some other things can impact it, like physical location, and this is especially going to be true for large populations that tend to be spread out over very geographically diverse regions. So these less than subtle geographical differences that occur very abruptly across a boundary are called clines, and they can actually cause variance within a population because that can possibly limit the amount of gene flow that occurs between members of that population. And if the amount of gene flow is severely limited, then you start to get to the point where you consider those members to be different populations, and you increase the possibility that we'll see speciation over time. Okay, so that is going to do it for this video. Thanks again for watching, and in our last video of this chapter, we're going to start talking about adaptive evolution. So when microevolution in a population occurs, what sorts of actual effects do we see on phenotypes over time? We're going to see that there's 3 different ways that a phenotype spectrum can change in a population over time. So thanks again, and I will see you in the next video.